brief CV with me ready. And so, Swapan, how are you doing? I,
Fine,
I,
fine. All right. All
yeah,
right. All right till
I,
now.
till now, yeah, yeah. But I noticed that you are very keen to get tested, huh?
Get what?
To get tested.
Oh yeah, yeah. No, not for me. That was not for me.
Okay, okay, okay,
Actually, my mother-in-law,
okay,
she's having a fever for 2 days, so
okay.
needs to be tested. And
Yeah,
now
yeah, yeah. Immediate.
any testing facility is taking 4 to 7 days. Imagine
Okay.
4 to 7 days just to
Oh,
get
that's the queue
test,
for testing.
no test results.
Oh, results. Okay,
Test
okay, okay,
results.
okay, okay.
So few number of machines, so few number of laboratories.
I
So
see.
so I was just searching here and there
Yeah, yeah, yeah,
where it can be
yeah,
done promptly.
yeah. Okay,
Mm.
okay, okay, okay. So I think uh, we'll start, Lata, right? And I mean, yeah, there are about
It
10
started. Going
students
join
who
one
have
minute.
joined, but I'm sure they will keep coming. They sometimes have many connectivity issues. So I have the pleasure of welcoming my college friend, Dr. Swapan Bhattacharya. Uh,
Good afternoon.
yeah. So let me just give you a brief. Children, he's from a very good school. I'm starting from his school. This is Ramakrishna Mission School in Calcutta. This is very famous. All brilliant students and illustrious students have passed out from this school. And then we met in Presidency College, Calcutta, which is also again very famous college. Now it is the university. In fact, we are from the same batch, and our batchmate is the vice chancellor of the university currently. Right? Yeah. Okay. And then Dr. Bhattacharya had botany as his subject in college. Children, you understand, no? Botany. Okay. By the way, I'll tell you, Swapan, I think I briefed you. These children are from class 9 from Parikrma Humanity Foundation. Okay. And then children, Dr. Bhattacharya actually did his master's in microbiology, specialized in microbiology, and then he did his PhD. We'll also request him, if time permits, to give us some brief about his research that he has done. Recovery of copper from ore by bacteria. That's very interesting. And then, of course, he has taught in many colleges for 36 years all across West Bengal. Right. And then he represented India in an international botanical congress in Vienna in 2005, Austria, right? And then since then because once you represent a country in an international congress, then you get invitations regularly from different countries, different forums. That's about his professional career, his domain. His expertise is, as I said, botany, microbiology, teaching. But he has also pursued, his hobby. His hobby has been writing poetry, reciting poetry. Drama has been his hobby. So children, are you all listening? Yes.
Yes,
And
Anna.
then and then he has published, I mean, his poems have been published, his short stories, his drama. He's been a stage actor, I know. And of course, he has written very popular science articles. So, this is a very brief introduction of our today's speaker, Dr. Swapan Bhattacharya. So, Lata, you'd like to talk about Parikrama to Lata is head of one of our. We got four branches. She's heading the Jayanagar branch. So, Lata, over to you.
Yeah.
Before we, yeah.
Good evening and a great pleasure to see you in our enrichment class. Students are waiting from yesterday and I gave brief about your about you. They're waiting for your class. And then Friday, Anna will be having quiz based on your session.
Yeah,
That's it.
actually, Swapan,
Hmm.
what normally happens is that we have a speak on Tuesday, and then I prepare a quiz based on that day's session and have it on Friday. And then so far I found that the performance, the outcome is very good, because they very minutely listen to you, and by the way, you will not notice them, but they're writing down notes while you speak.
Okay.
Yeah.
Okay.
So then, children, welcoming Dr. Swapan Bhattacharya, over to you.
Okay. Good evening. Good evening to all students.
Good
Good
evening,
evening,
Anna.
Anna.
Good evening, Anna.
Good evening to you all. Keep
And by
safe.
the way, I must tell you, they will address you as Anna. Huh? They are
Okay.
all Annas. Yeah.
Okay.
Okay. Yeah.
Keep well. Keep safe. At the very outset, I wish very good health and very good future for all of you.
Even we wish the same, Anna.
Thank you. Thank you. Uh, being class 9 students, you are perhaps aware of what is. I was thinking when Professor Bhaskar invited me to speak in front of you. I was thinking what I can talk about. Uh, this is a time where I tend to speak about the calamities we are facing, and I wish I could do that in very simple terms. But this is not a simple phenomenon. Uh, it is more difficult to put it in a simpler way. But to get a feel of that, I can delve into a few things which you perhaps are already aware of. You are, have you come across the term biotechnology? Have you come across the term biotechnology, students? I think
We
you
have
have.
heard it.
I think you already have. Of course, you are aware of the term vaccination by this time now. Although your age,
Yes, Anna.
you are aware of vaccination, and you know these vaccines which are being given right now against COVID infection are vaccines which are prepared by using technology which is precisely biotechnological techniques. Now, what is biotechnology? The term itself suggests that it is something to do with high-end machinery and complex some things, but not that complex, in the sense that biotechnology was being practiced before the time of our written history, because we know about, from the inscription of the walls, from the Egyptian mummy inscriptions, pyramid inscriptions, we know they were practicing brewing, they were practicing gardening, they were practicing agriculture. We all know the gardeners. Do they know about many things about biology or biotechnology? But how beautifully they produce flowers of different colors by using only their two hands, by pollinating one plant with the other, pollens from one stigma and putting it to the other. The development of the development of the high yield varieties of our crops, which were practiced even before the plant breeding or biotechnology uh, was known for any of us, or to any one of the human civilization. They were practicing these things because these are the things which are hidden in nature, The nature allows always permutation, combination, recombination, and that's how naturally varieties of flowers, varieties of crops, varieties of different fruits, etc., they come. When they come in nature, by either spontaneously or by the freaks of the nature. Initially, it was thought that it was a freak of nature. These were the freak of nature, that we sometimes we get a seedless papaya or seedless melon, or sometimes when we are keeping red and white red and white colored rose in our garden, we find suddenly rows of a white or pink variety, or rows of of another variety, a different color comes out. How it is possible? Yes, it was initially thought that it's just the freak of nature that these things are happening. But later, people, the agriculturists, the farmers, the gardeners, they began to practice these things. They began to practice by using their simpler hands, simpler tools. They began to practice these things and produced varieties, yielded cereals, yielded crops, which became far more useful, far more high-yielding varieties, far more useful for the entire humanity. So that is just that is just unveiling a property of nature which was there. Unveiling, just they began to unveil. Now, with the progress of our understanding of the intricacies of life, we came to know that the information of the life, that an offspring of human offspring will look like human, behave like human, will have characteristics, attributes of human, and an offspring of a cow, similarly, the offspring of a plant will behave like its own kind. So it will not exactly a clone, but it will be of the similar kind. It will be of similar kind. There can be variations. So no individual looks exactly like his father or mother, but their attributes are similar. Their characteristics are similar. They exhibit similar properties as their parents. So this is what we know as heredity. And after this heredity thing was known to the scientists, heredity was is known from a very long time. After this heredity was known, it was naturally the people were curious to know that where this heredity property lies, where in our body, or where in the body of a microscopic unicellular organism. All exhibit heredity. A paramecium divides to produce paramecium. A bacterium produce bacteria of similar kind. Similarly, a man reproduce an offspring which is similar to a man. So wherein actually these intricacies or these hereditary traits lie? And it was through some experiments from the beginning of 1900, that is 20th century, from 1926 onwards, 26 onwards, to 1950, the scientists came to know that it is something which is there in every living cell of our body, and similarly the body of every individual which is living. What is that? That is the DNA. That is the DNA. DNA, telling blanketly about DNA, or telling blanketly that it is only the DNA which bears the hereditary characteristics, will be wrong at the present time, because we know the virus which is causing havoc to us is not a DNA virus, it is RNA virus. So subsequently it was known that in some cases RNAs also can bear these hereditary traits. But it is, let us, for our understanding, we take it for granted that it is DNA wherein the hereditary traits lie. Where in the DNA? We call these genes. The term gene also came very long back. What is the gene? What is the gene matter? What is the gene? What is exactly the nature of the gene? It was through some milestone experiments from 1924, 1926 to 1950, 46, it was known to the scientists, or scientists came to know that it is the DNA where which bears these hereditary traits. And this DNA is passed on from one generation to another, bears the informations which is necessary for exhibiting the life's properties or attributes of life's properties from one generation to the other. Once this is known, the people began to think, well, if we know about DNA, why we cannot delve into this DNA thing? If we know the chemical characteristics of the DNA, then why cannot we just pick up, choose to cut a DNA, change a DNA, change its formation, change its chemical nature to bring about a result which goes in our favor? So from the basic understanding that this is done in the nature spontaneously or without manipulation, people began to think that yes, once we know exactly about the chemical nature, know about the way they are formed, their chemical characteristics, and the way they behave, why can't we just choose something which we know as the hereditary molecule and change it for our purpose? From that understanding, the biotechnology, the laboratory manipulation of the biotechnology began. From this very premises, the biotechnology became a subject which, at the molecular level or at the very basic level, at the very chemical level, at the level which you do not know, which you cannot see, but which an experimenter can direct. It is not that you are leaving everything on the freak of nature. You can direct the modifications. It is a modification which can be directed by the experimenter. Before that, can I share the screen with you?
Yes, Anna.
Yes. Can you see this? Can you see?
Yes.
Yes, sir.
Opening up. Yes.
Yes. Okay, I'm skipping this.
You can make it full screen also if you want.
I'm making it full screen. Trying to make it full screen. Sometimes it.
Yeah. I know there is a problem.
Sometimes
Yes.
there is a problem.
Yes.
Maybe there is a problem here also.
Okay, doesn't matter.
Can you see now? Is it full screen?
No, it is not full screen, but it is visible.
And
Acceptable.
down
Yeah.
you will be having the slideshow now. Icon you can use.
It is in the slideshow mode now.
Yeah, I normally what I do is I go at the bottom on the right hand. You see that 75% near, just on the left of 75%. I click
Next
that.
to that.
Okay.
Yeah.
Okay.
No, not that. You don't change that. You can keep it in the middle. On your left of 100%. On your left, yeah. There is another screen. Small screen,
No, I don't find
just
it
next to 100%. You can see 100% below. Yeah, just next. Yes, that one. You click that.
Can you see this now?
No, but I think there is something wrong, but doesn't matter. You carry on. We can see. Yeah. Role of bacteria in technology.
Okay.
In fact, 1 minute. You can make it a bit smaller, then we'll see the full slide. Little small. Yeah, correct. I think this is best.
Okay.
Yeah.
Fine.
So children, all of you can see?
Yes, Anna.
Students? Yeah. Okay.
You can see now. Okay. I'll come to this point later. Let us see. These are the two gentlemen, James Dewey Watson and Francis Crick, who actually discovered the structure of the DNA. This is here. They are standing with their model of DNA. Do you know What is the structure of DNA? What DNA is contained of? Do you know?
No.
Acha. For our simple understanding, let us say that DNA is like two strings wound around one another. DNA is like two strings wound around one another in a manner so that these two strings, one is anti-parallel to the other, not parallel, one is anti-parallel to the other. Anti-parallel means DNA is consisting of 4 bases. Do you know about acid and bases?
Yes, Anna.
Yes,
As
Anna.
bases, these are 4 bases. A for adenine, T for thymine, G for guanine, and C for cytosine. You just remember ATGC. ATGC are the 4 bases which are joined with one another in an anti-parallel mode. So naturally the question comes, if this is the genetic material, if this is the material which goes from one generation to the other, from parents to the babies, from individuals to their offsprings, what happens? It is first made half, then made 1/4 to the next generation, then 1/8 to the next generation. It is gradually becoming half, then 1/4, then 1/8, then 1/16. In this way, the things pass, this substance passes from one generation to other. If that is so, then in one entire molecule, the characters, the codes or the characters which are coded will be halved in the next generation. It will be half man. Then in the subsequent generation, it will be a 1/4 man. Then it will be 1/8 man. It is not possible. It is not logical either. So how the DNA divides is, there lies the key why it carries our hereditary properties from one generation to other, because they divide in a semi-conservative manner. That is, every time they divide, one of the strings are maintained and one of the strings is made anew. That's why all the characters which were coded by this molecule also are coded by these 2 molecules now. So each molecule, if each of these molecules go to one offspring, the offspring will be ideally like it was for its parent. So it is called a semi-conservative mode of replication. I'm not going into this is not necessary to remember, neither to understand the intricacies of this. Just remember that when the genetic substance, which is DNA, divides, it divides to transmit the hereditary characteristics, transmit the essential characteristics of life from one generation to other, not by dividing, but by replicating. Replicating is like forming a mirror image, not by dividing. If it divides, it will divide into half. But if it replicates, it will form a mirror image of its own. So by forming a mirror image, the characteristics are transmitted from one generation to other. No character is lost. That's why no character is lost. If a mirror image is formed here, then in the next subsequent generation, these 2 will form their own mirror images and pass on to the next generation. That way it happens. So the gentleman who gave this structure to us, these 2 scientists, James Dewey Watson and Francis Crick, they first gave the structure. Giving the structure or making us aware of the structure means making us aware of the substances or the molecules, chemical substances, by which it is composed. It is composed of the molecules. We now know, after their discovery, we knew about what they are actually composed of. And it was known that they are actually composed of 4 bases. You can imagine about just 4 currencies. You can shortly term them A, T, G, C. These 4 currencies. The nature has everything else on every of our cells are more or less monotonous structures. And nature brought about such a variation, 160 million types of lives in the world, all are composed of these 4 currencies, A, T, G, C, by only arranging them in different ways. So nature is a miser who brought about such variation in life forms, plants, animals, bacteria, viruses, etc., so on and so forth. Everything, all this entire variation in the life is brought about by expending only 4 chemical substances, A, T, G, C, by arranging them differently, and by maintaining the different sequences in different animals, different plants, different individuals. They bring about such a tremendous variation. So this is a huge miser's extremely open-handed expenditure for life. Miser is nature, but her expenditure for life forms are absolutely open-handed, created so many forms of life. There is one individual who was, this discovery was awarded 1951 Nobel Prize, the structure of DNA. This discovery was awarded Nobel Prize in 1951. Behind this discovery, there was a lady, Rosalind Franklin. She was the forgotten lady of DNA discovery. Just about her centenary has gone. She actually deciphered or found this structure. She was an X-ray crystallographist. That means with the help of X-ray, she can deduce or decipher the structure of different substances. Originally, she was doing with the structure of the coal crystals or crystals formed out of coal. So from this inorganic periphery, she went into organics and tried to X-ray DNA to suggest that what its structure could be like. This structure famously known as double helix. Helical structure wound around one another. Very well very easily compared with the helical staircase. You can compare this with helical staircase. You have seen helical staircases? Helical staircases, which... Have you seen helical staircases?
Usually with the fire exit.
Yeah, like fire exit, high-rise fire exits or these things. Helical staircases with a central axis and the steps wounding around this central axis. These are helical staircases. This is like 2 helical staircases wound around one another. From her research, this thing came. And as she was doing with X-ray, she gave the primary cue to what its structure could be. And subsequently, in King's College London, Watson, Crick, and her mentor, Franklin, Rosalind Franklin's mentor, they were awarded Nobel Prize, and she was forgotten. She ultimately died of ovarian cancer due to, as she was handling X-ray for such a long time. But she is the lady who first gave cue to this structure. Anyway, once the structure was known, then the question came how it acts, how this structure acts. You are talking about something which is hereditary. So right from your birth to right from formation of an embryo to death of an individual, everything, every living characteristic which one individual exhibits are coded by this simple chemical structure, for which the nature has only used 4 currencies. How is it possible? How do they act? Actually, it was known by that time that this DNA thing, this acts not by itself coding or directing some characteristics, not itself completely directing some characteristics or some behavior to take place. It just produces some proteins. So protein is the key substance which actually expresses right from our, say, hair color, right from our color of our eye, right from the way we talk, we behave, we think, we express ourselves. Everything is coded by, or everything is guided by one protein or other. These proteins are fundamentally known as enzymes. Have you heard of the term enzymes?
Yes, Anna.
Enzymes are catalysts, chemical catalysts, biological catalysts. What are catalysts? Catalysts are substance which enhance it or enhance the rate of a chemical reaction. Enzymes are the chemical substance, not exactly enhance, but which catalyze some reaction. So, for example, I am black-haired. So, hair color from the very onset of this reaction to form a hair to the end of the reaction, say it's about 200 chemical reactions, as a result of which the hair color becomes black. And for each of these 200 reactions, there is one enzyme. For each of these reactions, someone living in England, a blonde-haired individual, he has a blonde hair. His hair is also a result of 200 chemical reactions. But somewhere or other in these chemical reactions, one or two enzymes are different, which result in formation of a blonde hair color instead of black hair color. So it is just bringing about the variations in the enzymes. The hereditary molecule expresses itself differently in different individuals. Just someone very in Paracelsus, before Aristotle even, told that every life is a combination of some chemical reactions, and how true that is. Every life is actually a combination of several chemical reactions. It is because of some chemical reactions is different in man than that of a cow. A cow is a cow and man is a man. In that of a plant is different from that of a man. That's why a plant is a plant and a man is a man. Similarly, one man's characteristics, attributes are different from another man's because these two men's some chemical reactions among them are different from one another. Sometimes chemical reactions go awry. Sometimes the chemical reactions go absolutely haywire, causing what we know as genetic defects. For example, individuals with thalassemia, individuals with sickle cell anemia, they are born with some deficiencies in their blood, some deficiencies in the hemoglobin, as a result of nothing but one enzyme going wrong compared to the normal individual. So this is how the molecule of life performs. And it was these two individuals, one of them is, we are very proud of him, Hargobind Khurana and Marshall Nirenberg. The question was how the same four bases as we are talking about, four currencies as we are talking about, A, T, G, C, how these four currencies in all different forms of life behave in different way. They first gave the cue that before that we knew that it is by formation, by coding for different types of enzymes, for different enzymes, or just by coding different proteins, they behave in different way. So they searched what the proteins are actually formed of. Proteins are actually formed of, you have heard about amino acids. Proteins are formed of amino acids. Have you heard of amino acids?
Yes, Anna.
The proteins are formed of amino acids. They search for variations in the amino acids, and first suggested that the bases which we are talking about, A, T, G, C, are the currencies which we are talking about. Each three of them form one amino acid, and when we get 20 amino acids, we get a protein. A protein is at a minimum is formed of 20 amino acids. So it is just a search for 20 different codes in this arrangement of DNA, 20 different codes. They were the first who suggested that these are called genetic codes, the codes which bear our genetic traits, our characteristics. They suggested and they proved that each genetic code, which actually forms each amino acid, and ultimately the amino acids are and the molecules which form the protein. So it is genetic, these four bases arranged in threes, arranged in triplets, bring about this variation. The triplet nature of the genetic code. Just remember that they are the pioneers in suggesting how the DNA molecule act. So once we know about the structure of the DNA, and then we know about the molecules which through which the DNA acts, actually the proteins or the enzymes, and once we know about the structures they are composed of amino acids, and once we know that what amino acids actually form one particular protein or one particular enzyme, we know that if we change one amino acid here and one amino acid there, we can bring about a change in the structure of a protein, and as a result we can bring about the change in the structure of an individual or manner in which an individual acts. So from genes to genetic code, once these were the secrets of these were known to us, the people, the experimenters, began to search for something by which they can, because these are molecules, you cannot use your scissors to cut a DNA here and cut a DNA there, cut one currency here, take out one currency from here, add one there. You cannot do that. These are chemical molecules, and these are, for this you need scissors, but you need scissors which are equally molecular in nature, which are molecular scissors. People began to search by which, or with the help of which, how can we manipulate the structure of this? How can we change this structure? How can we change this structure at our will? If we can change this structure, say, we can do many things. Just one suggestion, that every individual needs insulin for digestion of sugar, digestion of sugar. Every individual needs insulin. One diabetic patient who is insulin deficient needs insulin to be injected or takes as medicine. So people began to search if we can change, two ways we can change an individual in relation to his or her insulin uptake. Either you can change the gene which codes for the insulin uptake, which codes for insulin, which is a defective gene, and change the gene in a manner so that it suits you, or you can produce a factory of the insulin by using something where the human insulin gene will produce insulin in your laboratory. For formation of human insulin gene, you cannot harvest human organs or organs of other animals, cows or buffaloes at your will to produce insulin at your will. So what is the simpler way of thinking about it, so that you can make a laboratory which produces live insulin for you, you can do that. If you change the gene of a bacteria, or if you incorporate the insulin gene of human individual, of one human insulin gene in a bacteria, and allow that bacteria to grow in your laboratory, and that bacteria, if that bacteria produces insulin for you, then you don't have to kill animals, you don't have to synthetically produce animals, you can harvest insulin at your will if you continue to grow on this bacteria in your laboratory. Whether this is possible, this will only be possible if you have scissors through which you can cut a DNA, or you can cut the hereditary substance, and you can have a glue by which you can rejoin your genes of your will from different sources, from other sources, from foreign sources, into an individual. That's what is known as gene cloning. Perhaps you have heard the term gene cloning. I'm skipping this. Molecular scissor. It was search gives results. For scientific experiments, you need to have scientific experiments proceed in 3 steps. First, you need to have you have to have an hypothesis. You have nothing. You just have your brain. No experimentation, nothing. No laboratory is required. You just have to think in your brain that this might be this. The apple falls to the ground and doesn't go up because there might be something which is known as gravity, which pulls the apple towards the center of the earth and not towards the sun and not towards the sky. Similarly, that is the hypothesis. Subsequently, second step is experimentation to prove it. For biological, or for this kind of experiments, for this kind of hypothesis, you need experiments through which you can prove this. And third is inference. This experiment can be experiments in the laboratory, or can be mathematical experiments, mathematical conjectures, which can be proved mathematically. Newton's experiments were proved mathematically. Observation was proved mathematically. And observations can lead to experiments as well. It was observed by that time, microbiology was in its prime. It was observed that in bacteria, there is a property. All bacteria, there are viruses which kill bacteria. Viruses kill human beings, viruses kill plants, viruses kill other animals. There are viruses which are specific for bacteria, which kill bacteria. And it was observed that some bacteria do not die, even if the virus attacks them. Some bacteria do not die. The first hypothesis was 3-step. First is hypothesis, then is experiment, and third is inference, that this actually takes place. The hypothesis was there must be something in a bacterial cell which allows, or which acts as a scissor to cut the hereditary substance. Number 1. Number 2 is that there must be something which rejoins these cut substances. So if this molecular substance is cut, then there must be something which fixes, which acts as a glue to rejoin. And it was observed, it was found that there are indeed some enzymes in bacterial cells which cut the DNA, which by this time we know are the hereditary substances, and which are very monotonous structures, T, G, C, A, G, A. These are formed of just four bases when they are differently arranged. But there are indeed some enzymes, some substances, which act as molecular scissors to cleave them, to cut them, to inactivate them. The discovery was made by these 3 gentlemen, Arbour, Nathans, and Smith. 1970 to 1972, these experiments were done. In 1978, they were awarded Nobel Prize. So once this molecular Caesar was obtained, then they began the search for the others in different laboratories. They began the search for molecular glue. When you have Caesars, you can cut the DNA. So when you have a molecular Caesar, you can cut the DNA of a bacteria, as well as you can cut the DNA of human individual to cut just the insulin gene and to transfer it into a bacterial cell. That is the hypothesis that is theoretically possible now after the discovery of molecular Caesars. Subsequently, these are the subsequently, immediately after the discovery, different enzymes. These enzymes are known as restriction endonuclease. You must remember the name, restriction endonuclease. Restriction endonucleases are the enzymes which cut the DNA, which are used for cutting the DNA in biotechnology, And then molecular paste. Molecular paste is another enzyme, DNA ligase. In fact, molecular paste was discovered before the discovery of the molecular Caesars. Now, you have two vital ingredients. If you are a tailor, you have both the Caesars and the sewing machine to cut your clothes according to your will and sew them according to your will, according to the pattern you have chosen. This is the two vital. Again, these are also enzymes. These are also proteins. These are also coated by the hereditary substances. But these are the substances which are harnessed, which are harvested from the bacteria to see whether one can really cut and rejoin the DNA. For this, bacterial cells were used. What is the advantage of using bacteria? Advantage of using bacteria is in the bacterial cells, that first bacteria has a very short living time or very fast living time. In 20 minutes, they complete their life cycle. That is, they born, they grow, they mature, and they die within 20 minutes. So in a 20-minute cycle, they complete all the reactions, all the activities of a life required, of a life, what the life requires for a comparable individual. For a human being, it is, say, average 70 years. So a 70 years life's activity is completed in a bacteria in, say, 20 minutes. First. Secondly, because they grow very that fast, 20 minutes is one kind of bacteria, but not all bacteria. Not all bacteria divide in 20 minutes. And imagine one bacteria which divides in 20 minutes. One divides into 2 in 20 minutes, divides into 4 in 40 minutes, 8 in 60 minutes, 16 in 80 minutes. If this way, one bacteria goes on continuing dividing and redividing, then after 48 years, can you imagine what a bacteria which has an average weight of 10 to the power minus 14 grams, that is 0.140, then 1. That is the average weight of one bacterial cell. What could be the weight of this bacteria which goes on dividing in this manner for just for 48 hours? It would be 400 times that of Earth. It will be a huge ball in the solar system which will cover the sun itself, maybe. It will be 400 times that of Earth, but that doesn't happen because they are self-restricted. There is, for everything, for every growth, there are signals where the growth must stop. Similar is for the population growth. The population, they grow by population growth, grow in numbers, and till a threshold number is reached where it will maximize its potential for harnessing all the available nutrients in the available to them. After that, they will stop. So there will be nutrient dearth, there will be stress, so they will stop dividing. And so that's why they do not grow, or they do not divide, go on dividing continuously. Anyway, that's a different story. But if they go on dividing for, say, instead of 48 hours, say 24 hours, or say 8 hours, say 12 hours, then what huge mass of bacteria you can get. Imagine what huge mass of bacteria you can get. And if in every individual, every bacteria, you can transfer, not every bacteria, just in one bacteria, you can transfer one of your preferred genes, say insulin gene, and allow it to grow and allow it to express this insulin, then you have a fermenter in which you are continuously growing bacteria which are producing for you the insulin in that container. You just harvest them, package them, sell them in the market, save lives, save individuals who are deficient in their insulin activity. So that is, but when you do this, this is neither its own DNA nor a complete gene or complete genome or complete DNA of one individual. This is called a recombinant DNA. For this, you have used first the scissors through which you cut the DNA according to your will, and then you join the DNA, bring out your gene of interest. In this case, I'm talking about insulin gene. Bring out your gene of interest from a human DNA. Join it with the help of molecular glue, what is DNA ligase we have talked about, and then retransfer it into the bacteria and allow it to grow. It will grow, and if it expresses itself, it will produce insulin, and that's what happens. That's how it is done. This is how I'm not going into this. Whether this is just to show whether because you allow the chemical reactions to take place, you allow the molecular scissor to cut, you allow the molecular glue to rejoin, but you don't see this. You cannot see this in the lab, with the help of your naked eye. Neither you can do it under microscope. Neither you can do it under electron microscope. You have to do it chemically. So you have to see it chemically, whether this ligation has taken place. For this, again, we use chemical measures. This is a gel which is called agarose gel, through which the DNA fragments can pass, and we stain it with the help of a dye. And if you hold it under light, in this case UV light, ultraviolet light, then they will show bands. From these bands, you can identify wherein actually among these bands, so many number of fragments, wherein lies your gene of interest, wherein lies the success of your experiment. But that is not simple thing, as not that simple as simply it is told. For this, I can just show you this to give an idea. So many fragments. You have allowed the experiment to take place. You do not know whether in this fragments of DNA which are actually cleaved with the help of molecular scissors, wherein actually which one is actually your gene of interest. For this, you can use what is known as markers. It's complementary as a fragment. Using a radioactive dye or a fluorescent dye, you allow them to freely hybridize, to freely mix and match. The band which matches with the human insulin gene sequence will be there, will be fixed on a particular cellulose matrix, and rest can be washed out. By this, by locating this, you can pick out your gene of interest. But it will become very complex now. Anyway, so what do we do? What do we do now? By using the techniques of biotechnology, what we are enabled with? We are enabled with Human Genome Project. We can, by fragmenting, once the molecular seizures are available, the human genome or the entire human chromosome set is formed of 3 crores, 3 billion ATGC, 4 currencies, which are 3 billion in number in different sequences. To read these 3 billion codes, 3 billion currencies, one after the other, is to read the sequence of genes, or genes which a human body or human cell contains. Is it essential? Yes, it is essential, because it is essential to know wherein actually a particular human gene lies. This was made possible once these molecular seizures were made available to us. Now, after that, the human genome could be fragmented into very short fragments, read one after the other, and that's how all 3 crores, 3 billion currencies can be read now, and one now knows wherein actually particular gene lies. Now, how can you use that for your benefit? You can use that for your benefit by, say, one individual who is immunocompromised individual. Immunocompromised means suffers from infection, has no immunity against the infections. Severely immunocompromised baby. His or her white blood cells can be taken, and a gene, it is called adagene, a gene of a preferred gene, which will correct this mistake or correct this deficiency, can be inserted into the white blood cell. And these white blood cells can be transferred back into the individual, into the baby. He or she who was immunocompromised, who cannot prevent the infections, who suffered always from infections, can be resistant of that. This is called gene therapy. If you know the location of a particular gene, you can remove the gene, remove the bad gene, insert a good gene in place of that, and that is called a gene therapy technique. It was not possible to cut a bad gene and insert a good gene unless you have the molecular scissors or molecular glue. Similarly, formation of designer drugs. Biotechnology had been very useful. DNA in the courtroom. Why? Because, say, in the case of murder victim, there are 3 suspects, and near the victim's body, a droplet of a blood is found. From that droplet, you can collect the DNA, fragment it, and from the individuals, from the suspected individuals, collect the DNA sample, collect the blood, take out the DNA sample, then allow them to mix and match. The mix and match will tell you who among these 3 is actually the culprit. Similarly, the paternity questions in court are also settled by DNA hybridization technique or DNA by biotechnological techniques. So it had been a courtroom tool for a very long time now, and very efficient courtroom tool. In forms, you can insert moth-resistant. You can insert moth-resistant genes in cotton, and you have moth-resistant cottons. You can insert disease-resistant genes in rice, paddy, wheat, and you can have disease-resistant rice. You can have genes which allow the plants to grow very fast and insert them into rice DNA, paddy DNA, so that paddy matures instead of 90 days. A paddy crop matures now in 45 days. You can insert genes from a seedless fruit to a seeded fruit, and you can get seedless melons, seedless papaya, seedless grapes, all by using these two tools. One is a molecular scissor for you, and one is a molecular glue for you. Use them at your will, but not to create demons, but for the benefit of humanity. When you leave it to the chance, there is a chance. Of course, there is a chance that you make Frankensteins, you make demons instead of individuals or instead of organisms, which would be benefit to us. The basic ethics of biotechnological technique is use these techniques for the benefit of mankind, not for making bioterror substances, not biological weapons, not making individuals at your will. There may be individuals are made at their will. The individuals could be made. This is Dolly the sheep. Have you heard about Dolly the sheep? Have you heard about Dolly the sheep? You haven't.
No.
You haven't perhaps. Dolly is the first sheep whose cell or from whose ovum another sheep, her baby, her daughter was born, who is exactly similar to that of Dolly. This is the first clone of one living organism, first clone of one mammal. Yes, first clone of one mammal. No mammal is exactly similar to that of his parents. No mammal, neither us nor you nor any individual. No mammal is exactly similar because we are not clones. Exact clones. There are variations here and there which take place during the course of development, which take place. To stop this developmental errors or developmental variations is to allow an individual to be born exactly similar to that of his parent. And that's what was first done in England in 1990s. This is Dolly the sheep. Her daughter, Dolly, has died. Her daughter is living. And yes, she is producing her children too. This is one possibility. This can be done. So if this can be done, individuals exactly similar to individuals similar to their parents can be theoretically made possible in the laboratory, can be grown possibly in the laboratory. Possibly. Who knows whether they are being made in one country or other? Nobody knows. But ethically, experiments which go beyond the nature's will, which go absolutely make haywire the rules of the nature, are ultimately will not be beneficial for the humanity. The tools you have, you can use them for the benefit of the mankind. There are many benefits. Many benefits have already been harnessed, and many are there to be harnessed in future. So this is just to here. I'll stop. This is a long lecture. I can go on talking about these things for a very long time, but let us stop here and let us just think about what basic premises we started and where we have reached. The basic premises was this, that in nature creates some freaks. These are there in nature, and human being always thought that why can't we apply these freaks of the nature ourselves? Can't we apply these freaks at our own will for our own benefits? For this, we needed to know what are the molecules we exactly should look into. It was found after 1951 that it is actually the DNA which should be looked into. Then they thought that why can't we cut the DNA at our will and change it at our will? For this, molecular scissors and normal scissors would not do, because these are not clothes. So molecular scissors, molecular glues. Are there some molecular scissors and molecular glues in the nature? Yes, they are. Of course, they are. They are not in. For this molecular scissors, you won't find them in any other or kind of organism except for bacteria. So go and look into the bacteria for these molecular scissors. And it was found that yes, there are many. There are plenty of molecular scissors which cut the DNA. So use them to cut the DNA at your will. Then if you can cut the DNA inactivate them, then you can cut the DNA as well to pick out a particular gene of your interest from an assemblage of several genes. And that's what they did. And after picking out this particular gene, they used the molecular glue to join two kinds of DNA from two sources, one from bacteria and one from human individual. Transferred into bacteria and watched whether the bacteria could produce this substance, which is a substance of our interest. And yes, indeed, they found that the bacteria, although bacteria, they are producing substances which are harnessed from human beings, and which are beneficial for human beings. So that gave a huge possibility. That opened up a huge possibility for the mankind to change, alter, beneficiate the organisms, and a whole new green revolution, the whole new gene therapy, the whole new revolution in drugs, in drug manufacturing, etc., took place after these discoveries. And ultimately, maybe it is possible to produce individuals at one's own will. Producing plants at one's own will is, of course, possible. This is called tissue culture technique. I just show you. You can produce now plants in the laboratory. You do not need to go to the soil. You can very well produce a paddy field in your laboratory in test tubes. It is possible. This is called tissue culture technique. This is possible. Just your laboratory will be producing, can be a paddy field, tissue culture. So that's how through tissue culture, plants are coming. Plants are being produced at will. Example is so many grapes, so many melons, so many papayas using tissue culture techniques, and so many flowers, so many orchids. You don't need to go to hills to have plenty of orchids for your floral arrangements in a marriage ceremony. These are all tissue cultured orchids. So you can produce lives in the test tube. Producing lives in the test tube, producing animal lives, or producing human individuals in the test tube, it is not possible because of properties which the plants, because plants you have seen, plants can be cloned easily. You can graft, you can take one branch of a plant, say hibiscus plant, and plant it in the soil, and the hibiscus plant will grow. That is the property of the plant, not of the animals. So it is difficult. Till now, it is difficult for animals. Plant have the property which is known as totipotency. From anywhere, from any part of their body, they can produce the whole plant. That is called that is known as property, which is known as totipotency. We now, as for the animals, we now have totipotent cells at our disposal. But God forbid, nobody at will can can implant the life in that cultured tissue as it is possible in case of plants. So that's it for today. Ashkar,
Yes, yes.
yes, is it all
Uh,
right?
yeah, absolutely, absolutely. Yeah.
Hmm.
Some questions, quick questions. Because you see, like only thing, Dr. Swapan, we normally close at 6:30, but such interesting subject. You also enjoyed talking, and they also enjoyed listening. But I am only afraid that we were 30 of us.
Uh-huh.
So, but they have a lot of household work and all that. So gradually, I think we
Okay,
had to. Yeah, yeah.
they can send you questions in mail.
Yeah, yeah,
You can mail
yeah,
me.
yeah.
You can
But
mail me.
any quick questions? Bhavya, Divya, Deepika, any quick questions? Or you can, Lata, you can take the questions and send to me. I
Okay.
will consult Dr. Bhattacharya and get back to them. Okay,
I
and
will,
if required,
I
we'll
will
call
type
him again.
my contact number here. If anybody wants to send to my number directly, you can send it to me.
Yeah, yeah, yeah.
Okay.
Okay. Right.
Okay.
Thank you so much. Thank you so much.
I
Very
don't.
interesting discussion.
Uh,
Actually, I was making notes. I'm sure all the other students also doing that.
oh.
Very good. Very interesting. Actually, I got a lot of clarity because these are young class 9 students. But what is recombinant?
Hmm.
That clarity I got today.
Okay.
Yeah,
Okay.
yeah, yeah. Thank you very
Okay.
much. Thank you very much. Yeah, yeah, yeah.
Okay. Thank you. I
Yeah,
also I did enjoy talking to you, every one
yeah,
of you. Thank you. Thank
yeah,
you
yeah.
very much.
But
Thank you.
I'm sure there'll be a lot of questions.
Okay.
I will flood you with questions from them. Okay.
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 Thank you.
Thank you, Dr.
Thank you.
Sapan
Thank you.
Bhattacharya.
Thank you.
Okay.
Thank you, Anna.
Thank you, Anna.
Yes.
Thank you, Anna.
Thank you.